understand that. Normally, a alveolar space is an empty space. Correct? Now these RBCs are coming from this blood vessel which are in the septa into the alveolar space, and it will fill up the alveolus. Now these alveolus are filled up with the RBCs. Now remember that in a lobe of the lung we get millions of alveolus or acini. All of them will be filled up with the RBCs, red blood cells or erythrocytes. So what will happen and how it look like? So once the alveolar spaces are filled up with the RBCs, it will make the part of the lung solid. Not only solid, it will look red in color. So this entire lobe of the lung now is solid and red in color. That is why it is known as red hepatization. Now this particular phenomenon is an important second step for lobar pneumonia. But this red hepatization won't persist for a long time. It will not. What will happen after that? These RBCs will be lysed. They will be destroyed. And now from these blood vessels, lot of neutrophils will come into this alveolus. So what we are seeing now in this stage 3, neutrophils are replacing the RBC to occupy these alveolar spaces. Now all these alveolus throughout the lobe will be filled up with these neutrophils, which will be nothing but a exudate. Now as a result of this transition, we can see the change of color. Now from the red, now it will become grey in color, because the neutrophils, the exudate has a greyish yellowish color, and that will produce a color of the liver, and that time once again the liver is very solid like a liver, and at the same time it is grey in color. So this is known as grey hepatization. Hepatization means the lung look like a liver, or lung has a consistency of a liver. What happens after that? Recall the process of acute inflammation. These neutrophils, supposingly, they will kill all the bacteria those are present in that area. Now once the neutrophil has done their duty, now the macrophage will come in and replace these neutrophils. Now what is the function of this macrophage? These macrophages, they will completely clear the area. They will remove all the cell debris or the necrotic tissue, and this process is known as complete resolution. Now why I am using the word complete? I am using the word because the macrophage will do a very fantastic job here. This macrophage not only cleared the area but also restored the normal lung histology. It means that it will bring the lung to the shape before the infection, and that is why lobar pneumonia in its natural process can cause a resolution. Definitely nowadays we need to give some antibiotics to help in this process, to help macrophages, and this is the area I want you to highlight very well. So the question could be like this: which particular cell is responsible for complete resolution of the lobar pneumonia? And what is the answer? The answer is macrophage. So once again, what are the various stages of lobar pneumonia? Number 1 stage, stage 1 is congestion. Stage 2 is red hepatization. Stage 3 is grey hepatization. And what is the stage 4? Is complete resolution. Complete resolution of the normal histology of lungs. And what is the name of the cell who is responsible for this resolution? It is macrophage. It is macrophage. Now here are some pictures you need to see here. There is a gross appearance of the lung on your left hand side. You can see a white circle. If you look at this lung tissue, the upper portion of the lung tissue looks solid. And what is the color? The color is grey, little yellowish. But the lower portion of the lung is supposedly a normal lung. And you can see the clear distinction between the area of the lung which is solid and the normal lung on the left hand side and the lower portion. Now this is very important to understand how the lobar pneumonia look like on a gross morphological examination of the lung. Now suppose it is grey in color, and it is looking grey, of course. So in which stage this gross picture you can corroborate? What stage is this? Is it a congestion? No. Is it a red hepatization? No. Is it a grey hepatization? Yes. So this grey hepatization is actually acute inflammatory process. So this is a time the acute inflammation is taking place, which is due to the cell. What is the name of the cell? The neutrophil. Now I want you to look at the X-ray image of the lung who has lobar pneumonia. Now can you tell me from this picture which portion of the lung looks abnormal? Yes, you got it right. It is the right upper lobe of the lungs. It is looking completely white. And this is a part of the lung which is solid because of the exudate or RBC. And as a result of this thing, this is looking white. And this is how the X-ray image of the lobar pneumonia look like. If you look at the other portion of the lungs, which is completely fine, and that is a unique thing about the lobar pneumonia. It is only a lobe of the lung is involved. Now look at this microscopy. I hope that you can identify the microscopy, because we saw something like this in our regular lab examination and the lab practicals. So what is this picture showing? If you look at the picture, you are looking at alveolus. Is it alveolus? Yes. Now I hope that you can see the cursor and try to see what is the alveolar septa. All this orange color stuff you see here, they are basically the congested blood vessel in the septa. This orange stuff you see, which is basically outlining the alveolar septa, they are the congested blood vessel. And inside this alveolus, you can see plenty of neutrophils. So it is the neutrophil which is now filling up this entire alveolus. It is interesting to know that in a lobar pneumonia, the alveolus or acini are the prime target. Bronchus or the airway, most likely they are not involved at all. So it is the alveolus where you can see most of this infection present in the lobar pneumonia. And that is one point you have to remember.